Welcome back to the Talking Ketamine podcast. This is episode 60, Ketamine versus Colorectal Cancer.
Great to be here. Today we are stepping completely outside the realm of mental health. We are looking at a 2026 paper by Korkmaz and colleagues that asks a wild question. Could ketamine actually help fight cancer cells directly?
Imagine waking up from a complex tumor removal surgery and finding out that the very anesthetic used to keep you unconscious was, well, secretly fighting the cancer alongside your surgeons.
Yeah.
That is the incredible premise of our discussion today. We're looking at this fascinating laboratory study focused on human colorectal cancer cells.
Specifically, a very stubborn cell line known as HT29.
Right, the HT29 line. So our mission over the next few minutes is to really understand how an anesthetic, a drug we usually associate with numbing the body or treating depression, might actually be weaponized against one of the most prevalent malignancies in the world.
Yeah, and to really grasp the weight of this research, we have to bridge a pretty significant gap. I mean, you might be wondering how on earth we get from treating synapses in the brain to targeting cancer cells in the bowel.
It does sound like a massive leap.
It is, but the bridge connecting them is the perioperative period. That's that critical window of time right during and immediately after a surgical procedure.
Like if you are going in to have a tumor removed, you have an anesthesiologist right there at the head of the table.
Exactly.
Their job is to manage your pain, keep your vitals stable, and basically keep you under.
Right, and ketamine is an absolute staple in that environment. Anesthesiologists rely on it heavily during major cancer surgeries because it's this profoundly effective analgesic.
Meaning it blocks pain really well.
Yeah, it blocks pain incredibly well without suppressing breathing the way some other drugs might. But over the last few years, the broader medical community started noticing this pattern.
What kind of pattern?
Well, they noticed that the choice of anesthetic agent used to put a patient under might actually influence tumor cell dynamics.
Oh, wow.
Yeah. The drugs running through your IV might be doing a lot more than just numbing the surgical site. They are circulating everywhere, interacting with any microscopic cancer cells that might be left behind.
Wait, let me make sure you, the listener, can really picture this dynamic. It's like you hire a security guard for your business, and their only job is to stand at the front door and stop pain from entering the building.
That's a good way to put it.
But then later, you check the security cameras and realize that same guard is secretly in the back room, systematically dismantling a burglar's entire operation.
Right. We hired ketamine to block pain, but the data suggests it might be actively disrupting the tumor itself.
Which is wild.
It really is.
Yeah.
And the specific enemy the researchers targeted in this study is that HT29 colorectal cancer cell line we mentioned. Colorectal cancer is a massive global health crisis.
And notoriously difficult to cure completely, right?
Very much so. Even after a seemingly successful surgery where the surgeon removes the primary tumor, the recurrence and metastasis rates are just devastatingly high.
Because, well, surgery itself is inherently traumatic to the body. It puts you under immense physical stress, which can temporarily suppress your immune system.
Absolutely.
And just the physical act of removing a tumor can sometimes, dislodge microscopic cancer cells right into the bloodstream.
Which is exactly what makes that perioperative window so incredibly vulnerable. If those rogue cells find a new place to settle while your immune system is distracted, the cancer comes back.
Ah, I see.
So because of that high recurrence rate, oncologists and anesthesiologists are desperately searching for new therapies we can use actually during the surgery to stop the cancer from taking root again.
So if ketamine is circulating during surgery, what is it actually doing to these rogue cells? I mean, what did the 2026 study find?
They observed what is essentially a two-pronged attack. The first prong is stopping proliferation. The defining, most terrifying characteristic of cancer is its ability to multiply uncontrollably, right? It ignores all the body's normal stop signs.
Right, it just keeps growing.
So to test if ketamine could throw up a stop sign, the researchers ran a viability test in the lab. They took these HT29 cells and seeded them into tiny wells on a plastic plate, about 10,000 cells per well.
Okay.
They let them settle and attach for 24 hours, just creating a baseline. Then they introduced different concentrations of ketamine to these microscopic environments and waited another day to see what would happen.
I imagine the untreated cells just kept dividing and crowding the well.
Oh, they did. In the untreated control group, the cancer absolutely thrived. But in the wells treated with ketamine, the rapid multiplication slowed down dramatically.
Really?
Yeah, in what we call a dose-dependent manner. As the concentration of ketamine increased, the survival rate of the cancer cells plummeted.
Wow.
At the highest dose they tested, only about 17% of the cancer cells survived.
That's a massive drop.
It is. But the crucial metric they calculated here is called the IC50. That is the exact concentration of a drug needed to wipe out half of the target cells.
And for you listening, the number they found was incredibly low, around 1.05 micromolar.
Exactly.
Now, 1.05 micromolar sounds like a microscopic drop in a massive bucket. It's hard to conceptualize just how small a micromolar is. Does that tiny amount actually translate to a potent defense in the world of pharmacology?
It is highly potent. A concentration of 1.05 micromolar is a very low biologically relevant dose.
No, okay.
To put it in perspective, that IC50 value rivals, and in some cases is even lower than, the concentrations required for several traditional heavy-duty chemotherapy agents used in colorectal cancer models.
Wait. Wow.
Yeah, the drug isn't just mildly irritating the cancer cells, it is neutralizing them efficiently at surprisingly low doses.
Okay, so stopping the cells from multiplying is fantastic, but if they were just sitting there in suspended animation, they're still a threat, aren't they?
They are. Which brings us to the second prong of this attack, what actually clears them out. It triggers something called apoptosis.
Right.
Now, cancer cells are dangerous because they essentially forget how to die. They bypass the body's natural expiration dates. But this study found that ketamine forcefully reminds them.
So apoptosis is programmed cell suicide, right?
Exactly. It is a highly orchestrated natural process that healthy cells undergo when they are damaged, infected, or simply too old.
But cancer cells mutate to shut off that self-destruct sequence.
Precisely. But the researchers used lasers and specific fluorescent dyes to illuminate the internal state of these cells. When they exposed the cancer to just that tiny 1 micromolar dose of ketamine, the lasers revealed that nearly a quarter of the cells had instantly gone into early apoptosis.
Nearly 24% at just 1 micromolar.
Yeah.
It is really important to clarify the difference between apoptosis and necrosis for you listening, because the study makes a very specific point about this.
It does.
Apoptosis is like a cell, politely packing up its desk, shredding its sensitive documents, turning off the lights, and leaving the building.
I like that analogy.
It's a clean, programmed, controlled exit that doesn't disturb the neighbors. Necrosis, on the other hand, is like the cell setting its entire office on fire.
Yes.
It is a messy, uncontrolled cell death that bursts open and causes massive damage to the surrounding area.
And the problem with the office fire necrosis is that it causes severe inflammation in the surrounding tissue. In the world of oncology, inflammation is like throwing gasoline on a fire.
Right, tumors love an inflammatory environment.
They thrive on it. It brings in blood flow and creates a fertile ground for more cancer to grow. A controlled apoptotic cell death avoids sounding that inflammatory alarm.
And the studies show that ketamine triggers that polite, controlled exit. The necrosis rate stayed under 1%, didn't it?
Exactly. Under 1% necrosis. It's incredibly clean.
So if ketamine is effectively forcing these gut cancer cells to stop growing and shut themselves down, we need to look under the hood at the mechanics.
Right.
How is an anesthetic, a drug, famous for interacting with the nervous system, achieving this inside a cancer cell that originated in the bowel?
Well, the researchers became molecular detectives to figure this out. They utilized advanced molecular docking simulations.
Which are basically complex computer models.
Yeah, you can think of them as incredibly complex 3D jigsaw puzzle simulations run on high-powered computers. They take the 3D chemical structure of the ketamine molecule and test how it physically fits into various target proteins on the surface of the cancer cell.
And they used multiple independent software models to make sure they weren't just getting a fluke result, right? They wanted to see exactly where the ketamine was parking itself.
Exactly. Both independent models confirmed the exact same primary target, and it showed a very strong binding affinity. The ketamine molecule was plugging right into the NMDA receptor on the surface of the colorectal cancer cell.
Wait, let's pause here. The NMDA receptor is a famous structure in neuroscience.
It is.
It is the exact same receptor that ketamine targets in the brain to produce its anesthetic, dissociative, and antidepressant effects. Why on earth does a colon cancer cell have a brain receptor?
I know it sounds crazy, but it speaks to the deeply opportunistic nature of cancer.
Okay.
Cancer doesn't typically invent entirely new biological machinery to survive. It is a scavenger. It hijacks the systems that already exist in the human genome.
Right.
So while NMDA receptors are most famous for their role in the brain, colorectal cancer cells frequently express these receptors on their surface and hijack them to fuel their own runaway growth.
So they basically steal a communication tower meant for the nervous system and use it to broadcast their own survival signals.
That's a perfect way to visualize it. And the specific signal they are broadcasting relies on calcium.
Calcium.
Yeah. Normally, the NMDA receptor acts like a highly secure turnstile that lets calcium flow to the cell. For a cancer cell, a flood of calcium is like a loud, blaring alarm bell screaming, "Stay alive, keep dividing, grow faster."
So what does ketamine do to that turnstile?
The computer models showed exactly how ketamine silences that alarm. It acts as an uncompetitive channel blocker.
Meaning it physically wedges itself in the way.
Exactly. It slips right into a water-repelling pocket inside the pore of the receptor. It forms a tight chemical bond, a hydrogen bond, with a specific amino acid inside the channel and completely plugs the hole.
Wow.
So the calcium physically cannot flow in. The alarm bell goes dead silent.
So ketamine acts like a genetic hacker. By blocking that calcium influx at the surface, it sets off a cascading domino effect deep inside the cell that hacks the cancer's core survival switches.
That's exactly right.
And the researchers actually looked at the genetics to prove this, didn't they?
They did. They ran an analysis on 29 different genes related to cancer signaling.
29.
Yeah. And they found 3 specific critical genes that were significantly turned down or downregulated by the presence of ketamine. The first is EGF, which stands for epidermal growth factor.
Okay, and a growth factor is exactly what it sounds like. It's a chemical messenger that tells cells to divide and spread.
Right.
When EGF is highly active, it gives the tumor the green light to invade surrounding tissue. So ketamine suppressed it, essentially taking away the cancer's engine and steering wheel.
Exactly. It cuts the engine. The second major target they found was a gene called TCF7.
TCF7, what does that one do?
This gene is a key player in something called the Wnt-beta-catenin pathway. You can think of this pathway as an ancient biological instruction manual.
Okay.
In embryos, it helps form the body's structure. But in colorectal cancer, this pathway gets hyperactivated. The cancer reads the instruction manual on a loop, driving tumor progression and metastasis.
So it's constantly telling itself to spread.
Yes. TCF7 is like the bookmark keeping the manual open. Ketamine significantly dialed down TCF7 expression, effectively closing the book.
Wow. If it's shutting down the growth factors and closing the instruction manual, it's systematically dismantling the cell's life support?
Yep.
Was the third gene?
The third was CSNK1D. This is a kinase.
A kinase.
Yeah, an enzyme that acts a bit like a cellular middle manager. Its job is to stabilize other proteins and make sure the survival signals keep firing efficiently across different pathways.
Oh, I see.
By downregulating this kinase, ketamine essentially fires the middle manager, causing the cancer's internal communication network to just fall apart.
It's a brilliant chain reaction.
Mm-hmm.
Ketamine doesn't need to dive deep into the cell and attack these survival genes one by one.
No, it doesn't.
It just cuts the main power cord at the NMDA receptor on the surface. Without that calcium flowing in, all those downstream survival systems, the EGF, the TCF7, the kinases, they all just power down.
Exactly. The cancer cell is left completely defenseless, which is why it finally triggers that polite apoptotic exit we talked about.
And the computational model supported this cascade perfectly, right?
Completely. When they simulated ketamine trying to bind directly to those downstream targets like EGF, the connection was very weak.
Sure.
The drug wasn't touching the genes directly at all.
It was all happening at the surface.
Right. Everything was happening because of that initial perfect blockade at the NMDA receptor. It is a cascading failure of the cancer cell's entire infrastructure.
The mechanics of this are absolutely fascinating. But, whenever we discuss a mechanism that shuts down cancer so elegantly, we really have to introduce a serious reality check.
We do.
It's very easy to hear all this and think we found a miracle cure. We need to ground this research so we aren't giving anyone listening false hope.
Right. I cannot emphasize this enough. This is an in vitro study.
Meaning it's outside the body.
Yes. That means the entire experiment was conducted in a highly controlled laboratory environment on a plastic plate. A petri dish is a vastly simplified environment. It is isolated. It is absolutely not a complex human tumor microenvironment.
Right. A real tumor in a human colon has its own blood supply. It is surrounded by immune cells, connective tissue, and this highly intricate three-dimensional architecture that protects it.
Exactly. So we simply cannot say that ketamine cures colon cancer in human beings based on a petri dish. It is a critical piece of foundational biology, but it is not a clinical trial showing that an IV drip of ketamine makes human tumors disappear.
That's an important distinction.
Very. However, within the confines of this specific puzzle, there is a fascinating nuance that researchers highlighted regarding the exact type of ketamine used.
Ah, the enantiomer nuance. This is where the chemistry gets really interesting.
It really does. So the researchers used what is called racemic ketamine, or RS ketamine. Molecules often have chirality, meaning they exist in two mirror image forms.
Like left and right hand.
Exactly. Think of your right hand and your left hand. They are mirror images, but you can't perfectly stack one on top of the other. Racemic ketamine is a 50-50 mixture of the right-handed and left-handed molecules. It is the most standard form used in clinical anesthesia worldwide.
But the body's receptors are highly specific shapes, like a glove.
Right.
And a left-handed glove only accepts a left-handed molecule.
And it is well documented in pharmacology that the left-handed version, known as S-ketamine, fits the NMDA receptor glove much better. It has a significantly higher binding affinity than the right-handed version. It holds on much tighter.
So if the entire chain reaction, the calcium blockade, the shutting down of the growth genes, the cell suicide, all depends on plugging that NMDA receptor, then isolating just the S-ketamine could theoretically amplify everything we just talked about.
Computationally and pharmacologically, that is a highly probable hypothesis. The researchers explicitly noted in the paper that enantiomer-specific investigations are the crucial next step.
That makes sense.
If you run this exact same test with pure S-ketamine, you might see even more profound rates of apoptosis at even lower doses, simply because the molecular fit is tighter.
Okay, so keeping the Petri dish limitations firmly in mind, while also acknowledging this undeniable cellular mechanism, how might this actually look in a hospital setting a decade from now? If we aren't using an anesthetic as a standalone cure, how do oncologists weaponize this knowledge?
It comes down to the concept of an adjuvant therapy. An adjuvant is something used alongside primary treatments to make them more effective. We are talking about using ketamine strategically to enhance conventional therapies.
Like pairing it with chemo.
Exactly. For instance, pairing it with standard chemotherapy drugs. If ketamine is dismantling the cell's survival pathways, it might make the cancer cells vastly more susceptible to the toxic effects of the chemo.
Or bringing it back to the operating room, using it specifically during the surgical removal of the tumor.
That is perhaps the most immediate application. During surgery, when those microscopic rogue cells are inevitably dislodged into the bloodstream, they are frantically looking for a new place to attach and grow.
Right, the most vulnerable time.
Yes. So if the anesthetic keeping the patient asleep is actively plugging their NMDA receptors, shutting down their growth factors, and forcing them into apoptosis, you are actively suppressing recurrence in real time.
Wow.
Preventing those micrometastases from surviving the trauma of surgery would be a massive clinical advantage.
It changes the entire paradigm of how we view anesthesia in oncology. You are no longer just putting a patient to sleep, you are deploying a chemical vanguard.
It's a huge shift in perspective.
Which leads me to a final provocative thought for you to ponder. The medical world spends billions of dollars every year trying to engineer new, exotic, highly complex cancer drugs. But what if the key to stopping metastasis has been hiding in plain sight? What if the simple choice of which anesthetic is used to put you to sleep for surgery could actually become our first, most immediate line of defense against cancer coming back?
It's a profound question.
It is wild to think a drug we rely on to heal the mind might also be able to tell a cancer cell to hit the self-destruct button.
It really is. It's early days, and a petri dish is not a human body, but it shows just how many secrets this molecule still holds.
A completely new frontier to explore. Thanks for listening to the Talking Ketamine podcast. We'll catch you on the next one.